Hey, welcome to chapter 4. So Robert Koch is often considered the father of germ theory. He was a German doctor who was also active in research. In particular, he wanted to study bacteria that were agents of disease, and after some failed attempts, he started to develop a medium that contained the nutrients needed, but he couldn't figure out a way to solidify the media. He first tried gelatin. And if you've ever made Jello gelatin, you know there are some general steps, and you finish with a liquid that eventually gets solidified into kind of like a polymer after it is refrigerated. Pathogenic bacteria prefer our body temperatures, and at our body temperature, gelatin would be liquefied. The other problem is gelatin is a protein, and a lot of bacteria can actually break this protein down and use it as a nutrient substance. So here comes the wife of his assistant in the late 1800s, assuming a homemaker or housewife, who offers the breakthrough that they needed. And it was to use agar, because she was familiar with how to use it when it came to hardening jellies. So by the 1900s, there's a major influx of study because now they're able to isolate and research many of these disease-causing bacteria, all by finding the proper ingredient and something that is going to be able to provide the proper environment and allow for growth of these bacteria, and they're not going to break down the ingredient that is keeping the media solid. Where are microorganisms found? First, remember that microbe refers to any microscopic organism, but it can also include viruses. Microbe doesn't just focus on bacteria, it also can be fungi, so it can be living or non-living. They can be found in the highest altitudes. They can be found at the deepest depths of the ocean where it's very cold and has an incredible amount of pressure. They can be found in the coldest regions or in the hottest heat pockets. Because of this, saying that there is life on other planets is not a huge stretch when life can consist of a single-celled organism. And we know that these can survive and thrive and grow in these really extreme environments, maybe where there's no oxygen or really high levels of acids or things like that. So some can grow without oxygen, some can grow without light, some have very minimal nutritional needs, and we will learn that others can form dormant structures. So even if they are found in an environment that's too extreme for them, they won't necessarily be able to grow and multiply, but they can form this dormant structure so that they can be kept safe until they find themselves in a more hospitable environment. And because they're found in so many vast environments, sometimes growing them in a lab can be tricky, but incredibly important. So we still don't know enough about these microbes, and some may have impactful significance in terms of cures or treatments or other medical discoveries, not to mention how we can utilize these microbes in industry. So first, being able to grow them, we have to first understand where they're normally found and if we can provide them with those conditions in a lab so that we can study them that way. Prokaryotic cells divide, but they divide differently. So it's not the same way eukaryotic cells divide. Our cells and other eukaryotic cells will divide through mitosis or meiosis, either one of these processes they're really reserved just for the eukaryotic cells. But prokaryotic cells, which remember include bacteria and archaea, they divide by binary fission. The cell will first increase in size, the DNA inside is replicated, and then the cell simply pinches off into two. So it's much more simplistic and faster and doesn't require as many steps and checkpoints that mitosis does, because mitosis, like I said, is reserved for eukaryotic cells, and as you know, eukaryotic cells are much more complex. It's not just DNA or cytoplasm, it's not just some ribosomes that they need to make sure are divvied out. There are organelles that require replication as well to ensure that each daughter cell gets everything that they need. The growth of cells increase exponentially because it doesn't just go from one cell to two cells to three cells to four. It goes from 1 to 2 to 4 to 8 to 16 to 32 to 64 and so on and so forth. So you end up with these huge jumps in population growth. And in the next slide or two, we're going to hear how to calculate that. And when we calculate it, you can really see the impact of this exponential growth, because you can start off with very little cells and in a very short amount of time end up with a very troubling amount that can have an impact in terms of health and disease. Each species will have a unique growth time, and the growth time is the amount of time it takes for that population to double. So the amount of time it takes to replicate what's needed in the cell and pinch off. Some species will be very fast. E. coli is one of them. It'll only take 20 minutes to replicate, and in that 20 minutes it is increasing exponentially. And other species can be much slower. All bacteria have an optimal growth rate, and this is the rate in which they will replicate the fastest because it is at a certain temperature, a certain amount of nutrients. They're basically in their optimal environmental conditions, and that's when they can exhibit optimal growth rate. When you step outside of these optimal conditions, either higher temperature or lower temperature, this 20-minute growth time will fluctuate, and they may still continue to survive, but maybe that growth rate is now 40 minutes, and you can see that's a really big change just by stepping outside of those optimal environmental conditions. Now, if you step too far outside of those bounds, too high a temperature or too low a temperature, too little oxygen, the growth rate may not only stall, the cell may actually die or become completely dormant. So again, that optimal growth rate is only going to be found when it is within its optimal environmental conditions, and that will be the fastest possible growth rate for that particular organism. And again, different bacteria have a different growth rate, and they can have very different optimal environmental conditions. So there is a simple way to calculate this, and when we do this, there's an example in the book and we'll walk you through it. It's really eye-opening to understand just how fast this happens and just how important it is to, I guess, be vigilant for scenarios in which this could occur. So to calculate how many bacterial cells will be in a sample after a certain amount of time, that certain amount of time that has passed, the amount of cells that are going to be present there is going to be represented with the NT symbol. When you know how many cells you started out with, you can calculate this by plugging in the amount of cells you started with as the N0 number. And then you also have to know the growth time for that particular species. So again, this could change. But all in all, it's going to help you calculate the number of generations that have passed with the amount of cells you started with to tell you how many cells are in that population after a certain period of time. So in this example, I believe it's a little bit more written out in your book, but somebody has prepared a potato salad for a potluck picnic and doing the best job they can. There just so happens that 10 pathogenic cells did make its way into that potato salad. Now, this happens all the time. We are constantly bombarded with potential pathogens or opportunistic pathogens or even true pathogens, and our body can fight them off, especially if it's a really small amount of cells. So starting with 10 cells, that's not something unheard of for pathogenic or non-pathogenic bacteria. But we know that this potato salad is going to be at this picnic in the sun, so it's not going to be refrigerated. It's going to be exposed to temperatures in which we are quite comfortable, which means that's the same temperature in which human pathogens can thrive. So if we know that this person accidentally encountered 10 pathogenic cells that made its way into the potato salad, we already know then the N0 number. So you would plug that into that calculation. Now, if we know what the generation time for this particular species is, and in this case we do, we know that it has a 20-minute generation time. So again, that means every 20 minutes it is going to double, it's going to replicate, and it's going to increase, not double, it's going to increase exponentially. We know that if 4 hours go by, one division every 20 minutes, then we would have given the opportunity for 12 generation times to have occurred in the 4 hours, right? Because in 1 hour, there are 3 20-minute blocks. 3 20-minute blocks times the 4 hours that are going by, that gives us number 12. So we're going to plug that in, but we're going to plug it in as the exponent for n. So that exponent is above the 2. So now we have 2 to the 12th power, and you can just multiply that, and that should give you 4,096. That's not our number, okay, because we started with 10 cells. So those 10 cells, we're going to now multiply that by 4,096, which means in 4 short hours, that potato salad went from 10 potentially pathogenic cells to 40,960 potentially pathogenic cells. That can be a really big problem. So what we're going to find out in later chapters is that each pathogen or opportunistic pathogen, they also have a threshold as to how many cells would have to enter your body in order for it to actually cause disease. Now, not many of them are ever going to be able to cause disease with just 10 cells. You know, there's some that require thousands, but 40,000 is a significant amount, and you have a really, really high chance of then consuming this and then feeling sick, you know, several hours later or the next day. So again, 4 hours is not a very long picnic. I'm sure there's plenty of times where you've gone to events where there is some sort of buffet, or if you ever started out with appetizers at your table and then had your dinner and then decided to pick again at those appetizers, it could have stayed out there for an hour or two while you're enjoying company. You have to take into account that everything, all of the food that we have and that we consume, will automatically be naturally colonized with bacteria. Okay, even if we boil it, by the time it's cooling, we know that there's environmental bacteria constantly floating in the air. They're making their way into our food and our water and drinks and stuff. So think about that next time, and maybe consider before you pick at cold food that's been sitting out there for a while, maybe to reheat it or pass up on that next time. Through the majority of microbiology history, we've studied microorganisms in laboratories. And of course, I mean, that seems like a logical thing. We can control the environment, we can control the lights, right? We're not subject to daylight savings time or anything like that. And, we can control that environment. But that's also the problem. It's a static environment. Even though, yes, you can change up that environment, right? We can lower the thermostat a little bit. We can turn on or off the lights. But compared to the natural environment, it's quite static. So think about how you behave in school. How do you behave in front of, when you were maybe in elementary and middle school? How did you behave in front of your teachers? Versus how did you behave in front of your friends? Versus how did you behave in front of your mom's friends whenever they visited? So if you think about bacteria as people and think about how your behavior changes depending on your environment, you can imagine that your behavior is going to be quite different depending on that environment. So the same thing with bacteria. In the lab, we have a very clean environment. They have little to no competing organisms. They may have competition amongst the same species, but really not the same that they're going to find in the natural environment. They are grown peacefully in media that is providing everything they could possibly need. So, is that anywhere near the same environmental conditions that you would find in nature? Again, if you have siblings, how do your siblings and you behave in different environments? And it can go from a camaraderie, and it can shift very quickly to competition. Same thing, right? The environment changes, and that matters. It makes a big difference in our behavior. Like I mentioned in previous chapters, all of us, including microorganisms, will try to conserve energy and not waste ATP. That means that if they don't need to make a particular amino acid because it's already provided for them as an ingredient in a media, they're not going to. If they don't need to express the necessary proteins to build a flagella or express and secrete the necessary polysaccharides to build a capsule, because the environment isn't requiring them to kick out their survival mechanisms, they're not going to. That also means that as we observe the bacteria in the lab, we aren't getting a true or complete picture of all of their capabilities because we aren't seeing them in the nature, in their natural environment when they do have to compete. So we have a very limited view and understanding because by researching them in the laboratory, we're narrowing the possibilities of competing environments. One of the survival tactics that bacteria will use, as we mentioned previously, is the ability to secrete polysaccharides that will form a capsule or a slime layer, and this helps them to adhere to things. Normally they wouldn't be able to, especially in fluid environments. They are subject to that environment. Even if they have a flagella, rushing water, they don't stand a chance to that. So they can go from this single cell floating planktonic bacteria to being able to adhere to things. And then after adhering, they can continue to secrete and build what's called an extracellular polymeric substance, and it forms a biofilm. So we mentioned this in a previous chapter. Again, if there is a free-floating planktonic bacteria and it's not able to attach to a surface because it cannot form a capsule, but a biofilm is already there, it can attach to this pre-made biofilm. So bacteria who aren't even able to produce this can benefit from biofilms. And it creates this really nice mixed community of microorganisms. You will see these biofilms often, like I said, in wet environments. And it's just as a necessity for the bacteria, for them to be able to survive. They need to create this to stick to a surface rather than being washed away. When bacteria are establishing these, they're actively multiplying and they're releasing a lot of these polysaccharides, which helps it to build, and it can build quickly. As they grow in this mixed community, the structure inside of the mixed community is not random or jumbled. So this is actually really incredible. It ends up being pretty coordinated. They form purposeful locations for bacteria in this biofilm, and that helps them to establish channels and pathways so that nutrients can easily pass into the biofilm and amongst that community. And it can also make it so that waste products can easily be shuttled towards the surface of that biofilm and away from that actively growing population. To me, that seems pretty incredible. Okay, you think about it. How often do you go to a store or you try to get to a counter to order something and people have to be told to form a single line? So, as adults, as humans, we have to be told how to stand in a line. But these single-celled organisms can create a somewhat organized community, and they can coordinate their efforts. And like I alluded to in a previous lecture, they can even communicate by sending out chemicals that will then bind to receptor proteins on other cells, and it lets them sense the environment, communicate, and respond to changes. And not just respond to changes as a single cell, they can actually respond to changes as a group. So this happens quite often in biofilms. They can exchange information as well, like information that can upgrade the cell, like those plasmids we talked about. And this is the perfect place for this to happen, since information can only be passed when the cells are in close proximity to each other. And they're definitely in close proximity with this biofilm. The other reason why this is really increased in a biofilm is it's not all the same species. If it's all the same species, they already have all the information. They either have the plasmids or they don't have the plasmids. Whereas in a mixed community, you're going to have some bacteria that do have plasmids and some that do not. But those that do not, have a really good opportunity of getting that information and being able to now be capable of those upgrades. Biofilms are also naturally resistant to a lot of things, unfortunately. Antibiotics, antimicrobials, detergents, and cleaners like Clorox. That's the reason why, the reason why is the nature of how thick that polymeric substance is. And they're all encased in it, and it just really protects them. An even bigger problem is that biofilms can grow on teeth. They can be found in lung mucus that accumulates in patients with cystic fibrosis. And as you know, people who have cystic fibrosis are already majorly immunocompromised. They can be found on medical instruments, medical devices, implants. You can imagine the amount of infections and damage that are caused by biofilms. Outside of the body, outside of human health, they can also grow in pipes, water towers, which is not only incredibly gross, but it can cause major economic damage to infrastructures and the economy. So again, can be a really big problem. So I just listed all of these really negative things that are attributed to biofilms. What I forgot to mention is they are resistant to detergents and disinfectants, and the only way to get rid of them is through mechanical means. So normally medical devices or instruments, they will be soaked in antiseptics and things like that. But if there is a biofilm that's there that probably can't be seen with the naked eye, we're not going to know that that detergent or antiseptic is not enough. You would have to physically scrub. And again, like I mentioned in previous slides, this is why if you were ever told to clean your shower growing up, you couldn't just sit there and spray bleach on the drain. It didn't actually do anything. You have to spray it and then you have to scrub it because that's the only way to remove it. Same thing with why we brush our teeth. The only way to remove the plaque, which are capsules and biofilms that have formed overnight or throughout the day, is to physically, mechanically brush it away. So again, really bad negative things for biofilms, but there's also some really good things that can come from them. If we can take microbes that naturally degrade harmful chemicals or break down pollutants, or the microbes that can consume and break down oils, won't they be even more effective if they can adhere and stick together and be applied, physically applied to the area in which we need them to concentrate? Now they can multiply easily. They are protected from things that can harm them, and they can be more effective in bioremediation. So again, a lot of negative connotations with biofilms, but there are ways in which we can use them to benefit us. Whenever you have microorganisms that grow in a mixed community, yes, we just talked about biofilm. I'm not talking about just biofilms anymore. Mixed community of microorganisms happen all the time. They have to be able to live and survive amongst different species. So look at your table, look at your floors, go outside and look at the soil. There's bacteria everywhere, and it's not just one type of species. So naturally in the environment, it's always going to be a mixed community, okay? And they have to be able to survive amongst very different species. And sometimes they grow in a cooperative fashion. They can actually help and benefit from each other. So you can have a bacteria that is using up and requiring a lot of oxygen, living next to a bacteria that actually cannot grow in the presence of oxygen. And because those oxygen levels are being lowered and diminished by bacteria number one, bacteria number two has a chance to live. So that would be one example in which they are surviving in a cooperative fashion. Maybe one bacteria will secrete a waste or byproduct that another bacteria can utilize. And so now the waste or byproduct is not accumulating next to the first bacteria because the second bacteria is taking that in and using it for its own means. So these type of interactions and cooperative relationships are really nice. They do occur, but they really don't occur as often as the opposite situation. More often, bacteria will be competitors. They are competing for the same things. They're competing for the same nutrients, for oxygen, for space even. And some bacteria will take it a step further than just this tug of war with nutrients or a shoving match. They'll take it a step further and they can get quite crafty. Some will produce toxins that can be secreted to kill and inhibit the growth of competing bacteria in its general vicinity. Some bacteria, particularly gram-negative bacteria where this is found, they will go so far as to be able to synthesize and use a needle-like structure that will protrude outside of their cell membrane, and they can use this to inject toxic chemicals into their neighboring bacteria. So this is definitely not State Farm. These are not good neighbors here. These are really diabolical neighbors. So this is a more directed attack, and it doesn't really leave anything up for chance. This is called a contact-dependent growth inhibition. So it's, again, goes beyond that shoving match to directed attacks. Like I mentioned in the previous slide, laboratory conditions really don't simulate the same environmental conditions. And so there may be more characteristics or capabilities of bacteria that we're not seeing. And I think it was already mentioned in another slide, but we can only grow and culture 1% of all of the known bacteria on this world. So there is 99%, really big number, of bacteria that for whatever reason we cannot grow them in a lab. This may be part of the problem. Maybe some species can only grow when they have the right neighbor, because they can only grow in a cooperative fashion. Oh, here we go. Here's that number 1%. My apologies for doing that spoiler alert. So how exactly are bacteria grown in laboratories? We already kind of know this because we've already met in lab by the time we get to chapter 4. I mentioned that it's not the same conditions as in the natural environment. They hopefully just don't happen to stumble across the lab. If there's bacteria growing in the lab, it's hopefully because we purposely put them there. They're usually supplied or grown as a pure culture. And a pure culture is a population of bacteria that basically originated from a single cell. So we know it's a pure culture. We know that it's just one type of species. And we know that all of the cells originated from one parent cell, and it was just given the opportunity to have, hours and hours and hours worth of division time. So there isn't competition. There's competition amongst themselves. Yes, they are going to need nutrients, but not the same level of competition that you would see in the mixed community. And all of the nutrients that they need, they're available for the cells. And it's not just any nutrients. It is nutrients that are needed for these particular cells in this particular pure culture. Without any competition of different species and with all of these nutrients, these cells definitely won't behave the same way that they would when there is competition. And like I said, only 1% of the known bacteria can be cultured and grown in a lab, and that's a really, really small amount. And there's really a lot of difficulty in trying to figure out how we can grow more, because 99% of bacteria not being able to be grown means that's 99% of bacteria that we can't research and we can't study and we can't figure things out. It's a lot of potentially beneficial research that we're missing out on. We know of a lot of these bacteria because we have DNA sequencing, but we don't know how they behave, and we don't know how they behave in a lab, and so on and so forth. So to ensure that you are growing a pure culture, you will need to minimize the potential for contamination. And as you have already learned, you're going to be using aseptic technique. You're going to be using that always throughout the entire semester, of course, because we never want to leave risk for contamination. As you know, these are the steps and procedures that should be taken anytime you're transferring bacteria, and it will greatly minimize the chances of contamination. It's not 100% because we have user error, but it's really going to minimize that chances. As long as you are doing it the proper way with the proper amount of time during each step, you should be fine. How do you grow them, or what exactly are you inoculating? So you inoculate a culture media, and this is something, it could be liquid or it could be solid, but it's going to contain all of the nutrients needed by your bacteria. And if you don't know what your bacteria is that you're using yet, it's going to have all of the nutrients needed for a really large majority of bacteria. And these nutrients are dissolved or mixed into a broth or solid media, and you're going to then inoculate it and you're going to allow it to grow. you will have propagated millions of cells of the same species that all arose from that single solitary starting cell. Now, one thing to know is if some bacteria have a 20-minute generation time and other bacteria have a much longer generation time, it stands to reason that you're not going to run to your incubator and check it after, 12 hours. I mean, you can, but if you don't see anything growing, you can't just write it off as an error. You need to allow for a couple of days because we do have the possibility of slow growing bacteria that do not have that 20-minute generation time. You don't want to throw away a petri dish before you've given an opportunity for those other bacteria that grow much, much slower. So we know how to grow a pure culture, but where are we getting that first sample from? Like, how do we get that first, um, pure sample from to then grow a pure culture or create a stock culture? Well, we first use a petri dish to allow for the space in which we can see the growth from a single cell. So it needs to have space so that they have room, and a single cell can then grow. And given the opportunity, it's going to grow millions of cells to form a colony. So you have the picture here of the petri dish. You can distinguish one colony dot from another. You can see that some are red and some are yellow. So I can visually see that this is two different species growing here. But more than that is the smaller dots towards the bottom of the picture. Let's look at the red one. Is much smaller than some of the dots that are kind of mushed together of the red dots towards the center of the picture. So towards the center of the picture, it looks kind of blobby right there because these cells were not spaced out enough so that when they did replicate and form a colony, it really overlapped and merged with another colony. You would then use your inoculating loop to aseptically transfer a sample from one of those colonies, the ones that we mentioned first, the smaller ones that are separate from a neighboring colony. And you're going to take it and now inoculate a sterile media that was provided for you. And then you're going to allow time for growth. So again, deciding which one to choose is important, and it may take a little bit of practice, but I think you guys will get it pretty fast. You're definitely not going to choose any quote-unquote colony that is humongous, because the reason why it got that big is it didn't arise from a single cell. It arose from several single cells that were pretty close to each other, and each started to grow a colony that merged into like a super colony. Yeah, it's all red, but it's definitely not from a single cell. So that's number one, our number one priority. And we can't say with 100% certainty that it even is a pure colony. So maybe it did arise from 4 cells. Is it possible that one of those 4 cells was not Serratia marquescens, which is the bacteria that has that characteristic red hue? Is it possible that there is one cell that is actually a different bacteria that is growing, but it's being kind of overtaken by the Serratia marquescens? We don't know, okay, because it's a pretty big blob. So some of that blob could actually be a different species. So if we were to choose that one, we can't say with 100% certainty that it is a pure colony, super colony. And we can't say with 100% certainty that we can create a pure culture from that. So just stay away from those types of options when you do this in the lab. In order for a colony to be visible to us, with the naked eye, there has already been growth to a threshold of 1 million cells. So that one cell that was isolated there replicated enough times to now have 1 million cells formed in that colony, which means you don't have to scoop and dig out that colony from the petri dish to get a sample. That would be way too many cells. That would be hundreds of thousands, if not millions of cells. You can literally use your inoculating loop aseptically, just lightly graze that colony. And by doing that, you're going to already have hundreds of cells on your loop. So then you use this and you are able to continue with your aseptic technique to inoculate your sterile media. The media that is used, like I said before, can either be liquid or solid. If it is solid, we know from our own experience in the lab that it can either be poured into a test tube or poured into a petri dish. But what makes it solid? Again, that ingredient we mentioned earlier, that agar that was used in the 1800s, we still use it. It is derived from seaweed. It is a polysaccharide, which should clue us into it being a carbohydrate, and it works really well, like I said before, because the majority of the bacteria don't break this macromolecule down. It can be heated to very high temperatures without the agar being degraded and like denatured or things like that. This allows for the possibility for sterilization. So we can sterilize media because agar can withstand the temperatures for sterilization, and it will liquefy. Another great feature is that after we've sterilized it and we've allowed it to cool, it won't solidify that quickly, which means you can let it cool enough so that it's a tolerable temperature for added nutrients, for other proteins, anything that's heat sensitive. It's not going to be a problem because the agar can be cooled enough without it being solid yet, so we can add those. We can even add bacteria before it solidifies. It will remain solid at a warm temperature, and when I say warm, I mean like the human body warm. And this becomes a really great type of media to grow pathogens on, again, because they do require and like the same temperature as what's in our bodies. So they do like warm-ish temperatures, and agar will not liquefy at these warm-ish temperatures. Gelatin, which was the previous ingredient of choice, it will. And as you know, a petri dish doesn't close as tightly as a test tube, but the fact that it has a cover does help to eliminate contaminants flying in. And again, we want to make sure that we always have that cover on unless we are actively inoculating the dish or a part of the dish. And we want to make sure that if we have lifted the cover, we're holding onto it and we're not placing it on our lab table. Because when we do place covers on the lab table, like a petri dish cover or a test tube cover, we have just stepped outside of those bounds of aseptic technique, and we have increased chances of contamination. So let's go back a step and discuss how to take a sample of that mixed culture, separate it from one bacterial species from another. We did this in the lab, so I'm going to just gloss over it pretty quickly. It's a specific method that is called the isolation streak plate method. Your book calls it streak plate. I prefer how the lab distinguishes it as isolation streak plate, so we'll go by that. By now you should have already experienced this in the lab by the time we get to chapter 4, but you're going to first take your petri dish, make sure that it's sterile, and draw a line so that you can divide it into sections. Your book, in this example, divides it into thirds, but in the lab experiment that we did, we have divided it into quadrants. So let's just say that you just took a sample from the dinner that your partner just made because you're pretty sure they gave everyone food poisoning. And being a good microbiologist, you're going to look into this. So you take a sample, but that sample obviously has a bunch of different microbes on it because, like we already said when we talked about the potato salad, bacteria is everywhere, floating in the air, and the minute we do something or make something, including food, bacteria will naturally settle into it. So it's going to have a bunch of different microbes on it. So you need to separate one from another so that you can take samples of single colonies, aseptically transfer that, and then look into if it's possible one of those are, the pathogen that's causing this food poisoning. So to do that, you take the sample and you're going to aseptically transfer it into that petri dish in which you have already divided into sections. And remember, the petri dish cover should always stay on until you're streaking, and then you should only open it partially when you're going to inoculate. So you open it partially, you inoculate once the first section, and then you'll close the petri dish, and then you sterilize your loop. You don't sterilize it and then grab another sample from the test tube in which you gathered a sample of the dinner, because you're just putting the same amount of bacteria on the next section of your petri dish. We don't want to do that. We want to thin out this sample. So you sterilize your loop, you allow it to cool. Again, you partially open your petri dish, and then you take, starting with your inoculating loop, from the first section and streak a sample of that into the second section. And then you're going to repeat this step to the third section and into the fourth section. And you just lessened the density of the cells that are found in that sample each time you did these steps. So that by the last section or the last quadrant, the cells have thinned so much that you can pick out single colonies, colonies that arose from an individual cell. You'll see them after a day or two, and then from there, you can do further steps like staining and then identify each of those colonies that you've thinned out and see if one of them is the culprit. Now that you have isolated a colony and you're grown a pure culture, now maybe you want to be able to maintain it. You want to be able to use it and refer back to it for maybe a week or something. Leaving it in the incubator is going to not be the option because eventually the bacteria will continue to grow, but they're also going to continue to use all of those nutrients, and you'll end up having dead cells there in the end. But we want live cells, right? So there comes time where we do want to try to maintain a stock culture, and we don't want them dying off. So one option that is relatively inexpensive is refrigeration. Taking the bacteria that, outside of the environment that promotes growth. So for mesophiles, we're going to be taking them out of the incubators, and then we're going to place them into colder temperatures that do not promote growth. And this will slow down the growth and kind of suspend those living cells there for a longer period of time than they would have normally in the warmer temperature. That seems pretty easy and straightforward. And then you can use the culture for a few, days, a week, or whatever, a couple weeks. If we want to try to maintain that stock culture for longer than just a few weeks, then another relatively inexpensive option is going to be just using a freezer. So that usually, goes hand in hand with refrigerators. But with this one, you want to make sure that you're able to achieve the proper temperature and maintain that proper temperature. So it may not be just a normal, personal home use type temperature that's found in freezers. And then you also have to add an ingredient of glycerol. And this is required because when you do transfer media from an incubator to a refrigerator and then to a freezer, or from the incubator straight to the freezer, you have a really high opportunity of ice crystals forming, and that of course is going to cause issue with the integrity of your stock culture. Now, the last option that your book mentions is not an option for us, but in other labs it is, and it can be, and that's freeze drying. So freeze drying, as the name sounds, utilizes both freezing and dehydrating, and again, it's an option if you have a really nice sophisticated lab. If you need to maintain this stock culture because you're working in some remote village and there's a strange disease going on and you're not sure how long it's going to take you to travel, and this and that and the other, chances are you don't have that equipment anyways, so this is just not an option for a lot of labs. But when you are working in a higher industry or in a government lab, this is going to be an option, and this is how they do maintain very, very old samples, and they can catalog those. All right, so let's think about this, how it occurs in the lab. We have isolated our sample, we found a single colony, and we created that pure culture. We have inoculated the petri dish or the test tube with that pure culture to establish it. What actually happens in that scenario is a particular trend of stages in the growth of these bacteria. What we have, our only option in our labs, is a closed system in which the media that's provided has a preset amount of nutrients. And as the bacteria grow, obviously they're consuming these nutrients, and as they consume nutrients, they are increasing the waste products, and eventually the nutrients in that media is going to run out. So because of that closed system, we will always see this type of growth curve. Again, it can shift somewhat depending on the species, but the general outline, the general shape is going to always be there. And these phases should also always be there. There are 5 distinct phases, and different things are happening at these different phases. So lag phase, log phase, stationary phase, death, and then prolonged decline or prolonged death phase. And we're going to just look into exactly what's occurring in each of these stages. So like I said, the first part is the lag phase. We have inoculated the bacteria onto the media, and there's no growth that's actually occurring here. They're not dividing yet. The amount of cells that are present here are the amount of cells in which you transferred. But the cells that are on the plate or the tube, they start using the nutrients there to synthesize enzymes and necessary chemicals that are required for replication. So during the lag phase, they are preparing for the step of replication. And the lag phase could be shorter or longer depending on the conditions, including the temperature, if it's correct for the particular optimal growth of that bacteria. Also the type of cells. Some may have a slower rate in which they will begin to replicate. Could be also the combination of nutrients if it's not ideal. So this lag phase can be shorter when we're at the optimum growth conditions, and then it can be longer for the same bacteria if it's not in the proper and optimal environment. The next phase, the log phase, this is when the cells will start to divide. The rate at which they divide will remain constant during this phase, and it's considered exponential. So we mentioned that before. During this phase of exponential growth, there are 2 sets of information that can be ascertained. So well, I guess three. One is understanding the generation time. So how long does it take for this particular bacteria to replicate and increase the numbers? This is really important if you're choosing a bacteria to do research on. You want to make sure that it doesn't have a very long growth time. Or if we're choosing this or researching this bacteria because we intend to use it in the production of something, maybe a medicine. The second thing that we can discover is the what are the primary metabolites? The primary metabolites are being produced at this time, and they are really useful to us. So these are amino acids and proteins that are being produced during the first section of the log phase. And we use those in industry. So if you've ever had protein or amino acid supplements or things like that, chances are they are being produced by bacteria, and this is where they would be produced and I guess harvested. During the second part of that log phase are secondary metabolites. So this is the third thing in which we can be discovering. Secondary metabolites are really important as well because what is being synthesized at this point in time by the bacteria are going to be antibiotics, molecules and chemicals that may be used as a means for competition, because towards the later stages of the log phase, they are going to start experiencing shortages, right? The number of bacteria are going up, so the space is decreasing and nutrients are decreasing, and this is when they're going to want to produce something that's going to inhibit those around them and kind of keep them in, with a safety net.
Once the level of available nutrients start to run low, continued growth no longer becomes an option. The bacterial count does not increase, but it's not quite at the point in which they are dying off. The count pretty much plateaus. This is because the few bacteria cells that are dying, and few being a relative term in comparison to the total amount of bacteria on the plate, they are releasing nutrients that can then be taken up by the few bacteria that are able to still divide. So in the end, net, the value or the amount of bacteria stays the same, because the same rate at which some are dying is the same rate that some are able to still divide. However, eventually the death phase is inevitable. So at some point, the number of bacteria dying starts to increase while the number dividing is no longer keeping up, and the cells start to divide at a constant rate. The rate is considered exponential, but not the same rate at which the cells were initially dividing during the log phase. So there is a downward slope. It's not as steep, it's not as angled as the upward slope of the log phase, but it's occurring. Many bacteria, but not all, would then show signs of prolonged decline. This is a part of the curve that starts to level off at the end. This is where some of the bacteria or microbes have survived the death phase. They have adapted. They've shown signs of adaptation that has allowed them to still function in an environment of diminished agar nutrients, surviving only on the nutrients that are being excreted by the dying cells. This environment is not conducive for normal or prolonged growth, which is why the curve just swoops. It doesn't spike back up. There's not a second log phase. However, if ever a species or strain of bacteria with extreme endurance or adaptation were to be discovered, it would be during this phase. The only bacteria that would live and thrive during this phase would have some major advantage over the strains that were killed off. This could indicate possibilities in terms of, super strains or super bugs or bacteria that can cause maybe unique infections. When you're growing bacteria in a lab, as we talked about before, they'll grow in colonies, and the colonies will have unique size, shape, and even coloring depending on the type of bacteria. These colonies are more noticeable on solid media than in liquid. And the cells that grow in a colony may show different characteristics depending on where they are found. For instance, cells growing on the edges or perimeter of a colony would have less competition for nutrients. There is a bit more space, the oxygen supply would be greater, and the cells have a higher chance of going through a log phase. This is compared to the bacteria cells that would be found in the center of a colony where there's more crowding, which means greater competition for nutrients, less available oxygen. These cells would be in a stationary phase, moving headlong into a death phase at that point. This means that primary metabolites would be found along the outer edging, while secondary metabolites would be found towards the center of the colonies. And depending on what a scientist is looking for, different parts of the colonies would be more useful. All of the previous details would be found in a closed system where nutrients are not being added. It's a set amount of nutrients from the beginning, and that's it. However, an open system would be the opposite. With an open system, you have nutrients that are continually added or added at regular intervals. This is great, but not ideal if the waste is not also removed. The buildup of toxins would still cause major issues. But in an open system, this is taken into account. Waste products are eliminated from the medium. This would require an input and an output source, which is seen in a chemostat. This device has a continual flow of nutrients in and a continual flow of waste out, keeping the cells in a permanent log phase, which would mean that there would be a permanent flow of primary metabolites. You can see that this system would be beneficial when wanting to study a uniform population with uniform characteristics, meaning there aren't these super survivalist cells being produced since there isn't a fight to the death for nutrients. This would also mean there is a constant and exponential production of primary metabolites, which would be extremely lucrative for the lab or for a pharmaceutical company. A great majority of bacteria live and grow in similar environments, the same that we do, or similar that we do. However, there are some that live in extreme conditions. These would be classified based on the temperature, atmosphere, pH, even water requirements or availability. So bacteria that grow in extreme temperatures will have proteins that won't denature or unravel in these temperatures. The proteins found in these microbes have a specific number of bonds and the specific position of bonds in the proteins that would strengthen or protect it from unraveling in these temperatures. The ideal temperature for each bacteria would allow it to experience a log phase. If the temperature rises or dips from this ideal environment, the production of necessary metabolites would slow. That means that refrigeration and freezing food would slow spoilage, slow the growth of bacteria, but not completely stop the growth, and certainly not kill bacteria. So in reference to bacteria that cause disease, the ideal temperature may be only a small window. This would mean that the bacteria would even be limited to infecting a human host depending on the part of the body. The skin on your hand is a different temperature than the skin behind your neck or underarm. And these are an even greater temperature difference from those found in your mouth or internal organs. That would mean some diseases have a very small window of where they can cause infection or damage. So there are 5 groups that bacteria will fall under in terms of temperature ranges. Again, if you're working with an unknown bacteria, figuring out what temperature allows your bacteria to grow most efficiently would help to narrow down your options of what you're actually looking at. So starting at the top would be the bacteria that thrive in extremely cold temperatures, followed by increasing temperature regions. Psychrophiles would be found close to the poles. Psychotrophs would be the type of bacteria that would survive well in refrigerated environments, which is how food items will spoil when refrigerated. Mesophiles thrive between the temperatures of 77 to 113 degrees Fahrenheit, generally the range in which we exist. The bacteria that cause disease thrive or are considered mesophiles. They thrive more specifically between the temperatures of 95 to 104 degrees Fahrenheit. This is where the human body temperature is generally maintained. So you could see how our temperatures and the temperatures that they need is like a perfect storm. Thermophiles we found in hot springs, probably hot tubs as well, and hyperthermophiles would be in temperatures of 158 degrees Fahrenheit or higher. Majority of the microbes found here are archaea. What's interesting is that microbes that require or use oxygen for respiration will actually produce a byproduct when oxygen is broken down. Now, obviously, byproducts occur when anything is used, such as waste, that's a byproduct. But these byproducts, called reactive oxygen species, ROS, are actually toxic to the cell. Not to other cells that aren't requiring oxygen necessarily, but to the actual cell that just underwent respiration. It's toxic to itself. Superoxide, hydrogen peroxide, these are examples of ROS, and they can cause major damage to the cell, even resulting in cell death if they aren't eliminated or altered. So the microbes that use or require oxygen for respiration also have to be able to produce superoxide dismutase. So this is a whole different other enzyme that would then push the ROS, the reactive oxygen species, whatever it is, into a chemical reaction that would convert these to plain old oxygen and water. So this is how they combat the toxic byproduct. Most bacteria also produce catalase, another enzyme. This one converts H2O again into, H2O2, hydrogen peroxide, into harmless water and oxygen. Microbes that are aerotolerant anaerobes. If you recall, these can grow in the presence of oxygen, but don't actually use it. They cannot produce catalase. So if you are working with an unknown bacteria and it has grown well on the surface of your media, if your deep tube or your slant or whatever, and you're not sure, is this an obligate aerobe or is it an aerotolerant? Well, one test you could use is just using plain old hydrogen peroxide. If you see bubbles, then you know that that cell has broken down hydrogen peroxide into harmless water and oxygen. The bubbles is the oxygen bubbling to the surface. If it does not do that, you have just basically narrowed it down that you're working with an aerotolerant anaerobe, or you're working with a cell that cannot produce catalase. So most likely an aerotolerant anaerobe. Bacteria control their internal pH the same way that we kind of control our internal temperatures. They control their internal pH with proton pumps. If you recall, pH is the measure of how many hydrogen ions are present. Okay? We talk about pH as in acidic and basic, but it's measured by hydrogen ions. The more hydrogen ions, the lower the pH value. The more acidic that item is, that solution, that cell, whatever, the more acidic. The less hydrogen ions, the higher the pH value, the more basic that item is. So pH is inversely proportional to the amount of hydrogen ions. When one goes up, the other one goes down. So if the internal environment of the cell becomes too basic, they would pump in, they being the cell, would pump in more hydrogen ions to then bring that pH slightly lower, increasing the acidity. If the internal environment of the cell becomes too acidic, they would pump hydrogen ions out of the cell, therefore raising the pH value so that it's more in that neutral range. Typical microbe would require a neutral or near neutral environment. This is why some foods add preservatives that are acidic. If you ever think about pickles or sauerkraut, these items have a long shelf life. Pickling is a form of food preservation because you're adding vinegar. All right, what's so great about vinegar? Well, the chemical compound of vinegar is acetic acid. Okay, it has a very low pH, and it actually protects the food from the majority of microbes. And since most of them are neutrophiles, they're not going to be able to grow in there. There are obviously exceptions. We already talked about H. pylori, right? This one grows and thrives in the acidic environment of your gastric lining, your gastric juices. Other exceptions would be those microbes that are classified as acidophiles. Acidophiles actually prefer or need a very low pH. And alkaliphiles, they will require a much higher pH, so requiring acidic or more basic. These are the ones that grow outside of that neutral or near neutral environment. Before, we talked about water and tonicity in class. We aren't the only ones that require water, but bacteria and microbes don't have the option to have crystal waters come deliver fresh water to them. So if their surroundings have a lot of salts or sugars, these molecules will bind to the water. So this is what we see when we are dissolving sugar in water. So let's say we wanted to make simple syrup, I don't know, or salt in water. We mix it around and it starts to dissolve. What's actually happening is that the water is binding with that molecule. So that water is no longer available. The salt or the sugar have hijacked them, basically. This goes back to tonicity and a hypertonic or a hypotonic environment. If the environment is hypertonic, that means there are a lot of solutes in the water outside the cell, meaning water, there's less water available outside of the cell. And in order for the tonicity to be balanced, water would flow out of this cell to bring about an equilibrium of solute to solvent ratio. This would dehydrate the cell, it would shrivel up, necessary processes would cease. This is another means of food preservation. Adding salts or sugars to food. This could include salt-cured meats. This is a way to prohibit the growth of microbes. Adding salt removes available water, and those microbes will then not be able to function because there's no water available to them, and now they're not only is there not extra water available to them, they're actually losing water to the environment. Adding salt and then removing water, so kind of, taking it a step further, like if you're making beef jerky, would ensure an even longer preservation. So of course, again, exceptions to every rule, halotolerant and halophiles can thrive on higher salt conditions. So there's still going to be some. So sunlight, carbon needs are combined in these groups listed here. So photoautotrophs would be basically highlighting the need for sunlight, and the carbon needs would be that is coming from CO2. So photoautotrophs. The way I remember it is auto, think of an automobile, which emits carbon gas. I think it's carbon monoxide, not dioxide, but work with me here. In the case of heterotrophs, carbon is coming from other mixed organic compounds, but it's still requiring energy from the sunlight. The final two terms mean that the energy is not coming from sunlight. So chemolithoautotrophs or chemoautotrophs, the energy is coming from inorganic compounds, and the carbon source is still coming from CO2. Whereas chemoorganoheterotrophs or chemoheterotrophs, the energy and carbon needs are coming from organic compounds.
Obviously, we talked about media a lot and choosing between liquid and solid, or the space that petri dishes can allow for versus test tubes. But we also have to talk about those ingredients. So again, we talked about ingredients being something that the bacteria are going to require, and trying not to miss anything that's completely necessary. There's two general categories of media, and all types of media will fall into one of these categories. The first one is complex media. Complex media has a lot of different ingredients. They're going to have, extracts and juices from various sources, proteins, amino acids, a lot of ingredients that come from some unsavory sources like animals or animal secretions and things like that. A lot of times you will find peptones. That's a very common ingredient, and peptone is just a mixture of various amino acids and short peptides that are produced by some animal source, and they're usually the building blocks for a wide range of proteins. So this is really important for the growth of organisms. Vitamins and minerals can also added there. Inside complex media, you'll also find some important ingredients for fastidious bacteria, which I believe we already talked about. Bacteria that just require a really, really specific diet, basically. They cannot make a lot of things themselves. They need it provided for them. And sometimes that is going to be from like those unsavory sources I mentioned before, like blood or things like that. So we'll learn about chocolate agar and blood agar. Blood agar we'll actually be able to use in the lab, but they are considered complex media. And a blood agar, it's kind of self-explanatory, but chocolate agar is actually also blood, but it has been heated so that the red blood cells have been lysed already. And the reason why complex media is called complex is, even though we do know the ingredients that can be found there, we don't always know exactly the amounts or the type of ingredients. So yeah, it's going to have amino acids. But this batch may be different from the next batch in terms of which amino acids. And especially if you're thinking about media that has sources from animals, going into the nursing program, I'm sure you've heard of blood sugar content and things like that, cholesterol, things that can vary in the same person day to day. So you can imagine if you're getting sources from living things, those ingredients and the amounts and the types can also vary from day to day. So we know in general what the ingredients are, we just can't say exactly this one type of molecule in this exact amount, and that's why it's called complex. This is in opposition with chemically defined media. And so that's the second category, is media in which we know the exact amounts and the exact ingredients, and it is going to be the same every time we order that media. And it's because it's usually synthesized and made up of chemicals. So the ingredients are more synthetic than what you would find in complex media. This is especially beneficial when you are trying to do a set of experiments and maybe compare the growth patterns of different strains of the same bacteria, or whatever kind of experiments you want. Remember when we talked about in chapter 1, experiments have to be very specific so that they can be replicated. Well, you can't necessarily say everything is replicated if you cannot control the exact ingredients. So this can be very beneficial for those types of experiments. And a lot of times buffers are added to make sure that the pH, whatever it is not going to swing or change very much. This is a table in your book on page 104, and it's just a really nice comparison between some of the ingredients that we would find in a complex media, and what they have here as an example is nutrient broth, and a chemically defined media, which is the glucose salt broth. With the nutrient broth, it looks like there's less ingredients, but beef extract and peptone, that's really vague. Peptone, again, just means that it's a mixture of peptides and amino acids. And beef extract, that's just parts of beef that have been extracted and maybe mixed and pureed and put in. What exactly is in that? We don't know. So you can see, even though less things are listed, it's really deceiving, because within those, especially the top two, obviously, ingredients, there could be a whole list involved. And that's, really different from the chemically defined media, which looks like there's a lot more in. And in reality, it's probably a lot less, because everything is listed out precisely and not just vaguely added there. Now that we know the two main categories of media, let's talk about types of media that can show us different results, and we would use them depending on what it is that we're trying to find. And the two categories that we talk about are selective and differential. Selective media can be either complex or chemically defined, but the purpose in which we use it is there's an ingredient in the media that will inhibit the growth of some species of bacteria and allow and promote the growth of others. So this is really important when you have a mixed sample. And when you're taking samples from a patient, that's all you're getting is a mixed sample. So if we're getting a sample from a patient, we may want to inhibit the growth of bacteria that we know never ever cause disease. And that way, the only thing that is growing are the ones that can cause disease, and then we would then examine those. Your book talks about McConkey agar. Your book also mentions Thayer-Martin agar. And Thayer-Martin agar is a media that is really important when you are trying to ascertain whether your patient does have a Neisseria gonorrhea infection, because it contains several antibacterial, antimicrobial drugs that will inhibit a bunch of bacteria, but the majority of the gonorrhea strains are actually immune to it. So you're allowing for this already fastidious bacteria to not have competition, and you're allowing um, for it to grow better without you having to continually thin out the sample and thin out the sample and hope that what you're looking for grows. If you can already eliminate and kill a lot of unnecessary bacteria, it really helps to cut down the time it would take for you to diagnose your patient. McConkey agar is not as complicated as Thayer Martin, but it is selective, and it will select for gram-negative rods specifically, which means it's going to inhibit gram-positive bacteria. So when you have to learn these, and you're going to have to learn them in lab, you need to know the full name of them. You need to know the particular ingredient that is inhibiting and what it is inhibiting, and what is it selecting for. So crystal violet, when it's used in the presence of growing bacteria, will actually inhibit gram-positive bacteria. And McConkey agar also has bile salts. And anything that can grow in that environment of bile salts means that it can grow in your intestines. So anything that cannot grow in bile salts, it's going to not allow to grow, because we don't care about them anyways, right? We're looking for specifically intestinal bacteria. So let's already eliminate any non-intestinal bacteria. The other type of media that can be used for testing is differential media. And differential media basically contains some ingredient that some microbes can utilize. A lot of times it is fermentation, so it'll be, lactose, and whether or not the bacteria can ferment lactose, we will see a difference in the colony growth. Okay, so differential media does not inhibit anything. It allows for anything to grow. But what it's doing is highlighting a difference in the capabilities of some bacteria. So just like selective media, for differential media, you need to know, the name of the media and the ingredient that it is, testing for, and what the bacteria would have to do or produce in order to use that particular ingredient, and show some sort of different look to their colonies. The other one that's listed in your book is blood agar. Blood agar contains the blood from, I believe it's sheep, and in the blood are red blood cells, obviously. And what we're testing for when we use this particular differential media is the ability to lyse red blood cells. We're looking for hemolysis. And so the bacteria will grow, and the ones that do not have any capabilities for hemolysis, they'll just look like normal colonies. The ones that do have capabilities for hemolysis, we'll actually be able to differentiate between them, whether they have depending on their varying degrees of hemolysis. So we can differentiate between beta and alpha. Beta being complete lysis of those red blood cells, and the colonies will grow, and the media around those colonies will become clear because they've literally lysed all of the red blood cells, and now the media that's around where they're growing doesn't have any more. And then alpha hemolysis, which is an incomplete lysis of red blood cells, and those colonies will grow, and the media beneath it will have like a greenish tint to it, and it's a partial lysis. So obviously, we're not going to use blood agar when we're looking for intestinal bacteria. We're going to use blood agar when we are suspecting bacteria that have the ability for hemolysis. And this is just a visual representation in your book. If we had a mixed sample of A, B, C, D, E bacteria, and we were to inoculate a media that just has allows for growth for anything, we would see all of those bacteria one way or another on your plate. But if we were using a selective media, only some of them would grow. If we were using a differential media, all of them would grow, but some of them would look different. They would have a color change or some sort of change that would distinguish a difference in their abilities to utilize one of the ingredients in that media. There is the option of, there are some media that are selective and differential. They are both at the same time. And this is what that would look like. Some bacteria are growing, and of the some that have grown, a few or one may then show a difference in their colony appearance, indicating that they have the ability to utilize whatever ingredient that is making this differential.
So this is going back to oxygen requirements of bacteria and how you would then account for these in a lab setting. You would have to have control over the environment. And to do that, you would need, certain machinery. The picture on the left is a container that has a packet of compounds that would reduce the oxygen content. Content. Gas pack jar, it's called sometimes. Other options would be a chamber, the one on the right, much more sophisticated, probably, I'm assuming, much more expensive. Other options would be growing the bacteria in a media containing sodium thioglycolate, which is a reducing agent that just reduces the amount of oxygen in there. Enrichment cultures is exactly like it sounds. You are enriching the cultures with nutrients so that the bacteria may exist in bacteria that may exist in small numbers would then be allowed to flourish. This could also be compared to selective media, since some of the enriching ingredients would promote the growth of bacteria, but also it could it would not necessarily promote the growth of all the bacteria there. All right, so we have isolated the bacteria, we have promoted its growth, we grew our bacteria. How are we going to count them? How do we know how much bacteria colonies exist? One way is using a colony counter. You will see one in a lab. It's basically like a magnifying glass with a light shining and some grids. All it does is basically make it a little bit easier to count and keep track of the colonies you've counted. Another one that's pictured here is a cytometer. Uses a light source. It aims a laser at the culture. It's then measured as the culture then passes through the laser, and the amount of light that is scattered is proportional to the amount of cells present, because each cell would actually cause the laser beam to scatter and not pass through or be reflected. It'll refract the beam. Another method to detect and measure microbial growth would be counting only viable cells. So the ones that we just mentioned before, those are counting any cell that is found there. Now, not every cell will produce a colony. And so that's the difference here with this method. We're only going to count the cells that are capable of multiplying and producing a colony. But if you were to inoculate a plate from an original bacterial sample, as you can see with our past experiments, there are many colonies, way too many to count. So what you would do here would be a series of dilutions. You would take 1 milliliter from the original sample and put it into a next vial. And now fill that vial with 9 milliliters of sterile culture, or it could be of sterile water. So now you've diluted it by 10%, basically. Then you would take the second test tube, take 1 milliliter from that, put it into the third test tube, and then fill that with 9 more milliliters of a solution, or a solvent, whatever, either more broth or just sterile water. And you would continue to do this and then when you did that, you would then take from each sample and inoculate a petri dish and see what grows. Now, most likely, the original sample, the first dilution, and probably the second or third dilution are still going to produce way too many cells to count. But eventually you will find that one of your dilutions is producing viable colonies that can be easily counted, that are large enough to be considered an accurate statistic, but then just small enough number to be easily counted. And that usually falls between 30 and 300 cells. So once you do that and you've allowed the petri dishes to grow and you picked one that falls between 30 and 300 colonies, what you would then do would be to multiply the amount of colonies you counted by the amount of dilution. So if each one was 1/10 of a dilution, you would multiply it either by 10 or 100 or 1,000, 10,000. So in multiples of 10, depending on how far you had to dilute it. And then you would get not an absolute 1,000,000% accurate number of viable cells from the original sample, but you would get a very good general number of how many viable colony-producing cells were in your original sample. And I'm sure you can probably pause this and maybe click on that YouTube link at the bottom. That one as just a YouTube video I found that maybe might explain this a little bit better. Other methods would be doing a spread plate, which we did in class, and we streaked it multiple times. This is another method. It's a multi-step method. It involves first filtering your liquid media, and you're filtering it through this system that actually has like a paper filter at the bottom, I guess similar to a coffee filter. And so any cells would be caught in that. And then you would be able to actually take that paper, that filter, and place it with media and allow it to grow. And now you're able to see how many colonies were grown. So again, it's only counting viable cells, right? But again, it's multi-step, this one, and might be a little more cumbersome. This is similar to your serial dilution, and it's not really discussed. Here's another way to measure the amount of bacteria utilizing a spectrophotometer. Kind of similar to a cytometer. Again, it's not giving an exact number. It's more measuring the cell mass present. Again, it's not measuring viable cells. We don't know how many of these can actually form colonies. We just know that, that they're there. So they could be dead cells. We really don't know. But this uses a machine that shines light at a liquid sample. It's not a liquid sample that's being, run through the laser like the other one. This is just, we took, 1 milliliter or 10 milliliters of the original sample or something like that, put it in a small vial, stuck it in this machine. So again, it's not the full sample usually. And the light is then aimed at it and filtered through. The more light passing through would mean the less bacteria present. It's basically just measuring it's basically just measuring turbidity. Oh my gosh. Okay, so this is measuring turbidity or cloudiness of a sample. More or less how many cells are in there. Again, it's a general amount. It's not a specific number, and it's definitely not attuned to viable units or viable cells. And this is what it would look like then. So if, again, it's inversely proportional, you can measure the amount of light absorbed, which would then mean more growth. Or you can measure the amount of light passing through, which would mean an inversely proportional amount. And that's basically it for this, the rest of this chapter. So go ahead and run through this, and you can watch it as many times as you want. Please look at the study guide that I posted up there as well. Get to work on that. I will see you guys on Wednesday, and we will be kind of flying past, I think, chapter 5. I'm not sure, again, how much is actually going to be needed, but also you already have the study guide. So the topics that are listed there for chapter 5 are the ones that I do want you guys focusing on.